Hello, this is Dr. Barbara Oswald with our Module 2 overview for our PSS 312 Drugs and Behavior class. In Module 2, we learn how drugs work. By the end of this module, students should be able to describe at least 7 ways drugs affect neurotransmission. You'll get the chance to demonstrate your knowledge of this in discussion options 1 and 2 and homework questions 1 and 2. Then you'll learn to describe at least 7 major neurotransmitter systems in the brain, and then for each neurotransmitter system, be able to identify at least one drug that functions as an agonist in the system, and at least one that functions as an antagonist. And this will be assessed through homework question 2. Next, you will learn to identify at least 7 different routes of administration and explain how the route of administration impacts the potential for addiction. We'll cover this in discussion option 3 and homework question 3. Next, we'll learn to distinguish between pharmacokinetics and pharmacodynamics. You'll learn to describe at least 3 pharmacokinetic principles that impact drug effects, and this will be assessed through homework questions 4 and 5. Next, you'll learn to explain the therapeutic index and the margin of safety as a way of understanding drug toxicity. This will review in discussion option 4 and homework question 6. Next, you'll learn to explain the effects of learning on addiction, identifying and distinguishing between effects learned through classical conditioning and those learned through operant conditioning. And we have 3 homework questions about this, homework questions 7, 8, and 9. Finally, in this module, you'll learn to identify several methodology used to study drug effects and addiction in the laboratory. And this will cover in discussion options 5 and 7 in the Module 2 exam. So what work do you need to complete for Module 2? Well, in Module 2, you'll read 3 chapters, chapters 3, 4, and 5 in our assigned textbook. You'll read the Module 2 research article provided on Canvas. You'll take the practice quizzes over chapters 3, 4, and 5. You'll participate in the Module 2 discussion. You'll complete the Module 2 homework questions, and you'll complete the Module 2 exam. Remember, there's 2 parts to the exam, 25 multiple choice questions similar to the practice quizzes over the readings in the book, along with several essay questions based on the research article. So, in the few minutes that we have left in this screencast, I'd like to take some time and just give you a big overview of what we're going to cover in this module, and spending a little bit of time going over some of the more difficult concepts that students have in this module. So the module overall is trying to help us understand how drugs work in the body, so that we can understand how they exert their effects on behavior. So there's quite a bit of review in Chapter 4 from Psych 251, Introduction to Biopsychology, which is why that class is a prerequisite for this class. So your basic understanding of neurotransmission, that is how neurons send signals to each other using neurotransmitters, understanding concepts of agonism and antagonism, the idea that drugs can function to either increase or agonize natural neurotransmitters, and drugs can also antagonize or block natural neurotransmitters. So we'll review those principles of how that works in Chapter 4. Then we'll go over 7 major neurotransmitter systems, also a review from Psych 251. ACH is acetylcholine, DA is dopamine, NE is norepinephrine, 5-HT is serotonin, GABA is GABA. It's the abbreviation for gamma-aminobutyric acid, but we always just call it GABA, glutamate, and the endorphins. So this review is important because all psychoactive drugs, that is drugs that get into the brain and change behavior, they change behavior and consciousness by functioning as agonists or antagonists in these major neurotransmitter systems, as well as others. But so knowing what neurotransmitter a drug affects and knowing what that neurotransmitter does will tell you both the effects of the drug as well as its addiction potential. So for example, we know that dopamine affects reward. If you take a drug like cocaine or methamphetamine that has direct effects on dopamine, and they function as agonists, meaning they increase dopamine in the brain, they're going to have a really high addiction potential because of those effects on dopamine. So understanding that can really help you understand what drugs do and what effects they're going to have on behavior. So what also then gets covered in Chapter 4 is not review from Psych 251, but it really impacts what we learn in this class. So the first thing we talk about are pharmacokinetics, that is how drugs get processed through the body. We use this acronym called ADME, and that stands for absorption, distribution, metabolism, and excretion. How a drug gets absorbed into the body can really determine its addiction potential. Does it get absorbed directly into the bloodstream and go right into the brain? That can lead to really significant addiction potential because it provides instant gratification, as opposed to other drugs that might be taken orally, where they have to go through the stomach and then pass through the small and large intestines and then kind of leak out into the blood and then make it up into the brain, so that it takes maybe 45 minutes before you feel an effect of that drug. And that can really kind of decrease the addiction potential. It doesn't erase it, but it can explain in many ways how addictive a drug can be. The general rule of thumb is the faster into the brain, the quicker the high is experienced, the more addictive that drug will be. So we talk about how quickly it gets absorbed into the bloodstream and then into the brain, how the drug gets distributed throughout the body. Does it get stored in fat cells? That's going to affect, drug testing, which we talk about in this chapter, as well as how drugs get metabolized through the liver and then excreted out of the body. So that's one thing that we cover. As I mentioned, the pharmacokinetics, especially absorption and distribution, are really affected by route of administration. In this chapter, we're going to learn about 7 different routes of administration. This little infograph kind of illustrates a few of those, talking about, drugs can be taken orally, like in a pill form. Some drugs are taken rectally, inserted through the rectum. Some drugs are injected into muscle. Others are injected directly into a vein. Some are stuck under the skin, like skin popping. Ketamine is a drug that a lot of people like to take through this subcutaneous injection. Or transdermal would be like a nicotine or a fentanyl patch, can be absorbed through the skin. Inhalation, anything that gets smoked. We also have things that get snorted. That's not on this graphic, but you could snort things through the nose. And all of that determines how quickly it can get into the blood. Obviously, if you inject directly into a vein, it's immediately in blood and right up into the brain within just a few seconds. Inhalation is also quite quick. It goes into the lungs and then distributes very quickly to the heart, into the blood, and then into the brain fairly quickly as well. Same thing with snorting. And so we know that all these compounds that are either injected into the blood or smoked or snorted have a really high addiction potential because they cause these fast, intense highs. Some other things that I mentioned as well, that the pharmacokinetics, or absorption, distribution, metabolism, excretion, impacts drug testing. And so I know this isn't the greatest graphic, but we'll talk in this chapter about how some drugs can be detected in, let's say, saliva. Other drugs can be detected in urine. Other drugs can be detected in hair or other body cells for a certain number of days. And it all is based on the pharmacokinetics of the drug. So some drugs stick around in urine for quite a long time. For example, benzodiazepines. THC can be detected in urine for chronic users up to 30 days. things like that, whereas others will dissipate out of urine pretty quickly. Cocaine is only 3 days and so forth. So we will talk about pharmacokinetics of various drugs, both introducing the idea in this chapter and then investigating specific drugs more precisely in later chapters. So the chapter also talks about pharmacodynamics. Pharmacodynamics refers to how drugs then affect behavior, basically. So we talk about dose response or dose effect curves. And here we have a dose response, or I think your book calls these dose effect curves, where it's showing the dose of the drug, and they call it log doses. We usually give drugs in base log 10 doses, but just imagine that this is a very low dose of the drug, and up here would be a much higher dose of the drug. And in low doses, we have very little effect. Higher doses, we achieve a maximal effect, where it gets what we call to asymptote. It tops out. And then we have the ED50, or the effective dose 50% of the time. And so that's usually somewhere in the middle of these dose response curves. So we can talk about the ED50 and the LD50, or the lethal dose or toxic dose. So you can describe both sort of differently. So toxic effect might be where you're posing danger, but it's not deadly, or a lethal effect, as the name suggests, is the lethal dose. And so we talk about a drug's margin of safety, which is the difference between the effective dose, whoops, I didn't mean to jump up there yet, the effective dose, which might be here, and then the lethal dose. And then this would be the margin of safety. So for example, if the effective dose of morphine is 4 milligrams, it's actually 4 milligrams per kilogram, and this might represent 20 milligrams per kilogram. That means if a person took, for example, instead of 1 or 2 pills, maybe took 4 or 5 pills, they run the risk of overdose. So understanding this relationship between the ED50 and the TD, toxic dose, or LD50, lethal dose, can really help us understand why some drugs are so prone to overdose while other drugs are not. And so again, that understanding leads us to what's called the therapeutic index. And you can calculate mathematically the therapeutic index by dividing the toxic or lethal dose by the effective dose. And then you would know exactly where the danger might run. And so this might be actually probably represents heroin. So it's a pretty low dose of heroin that would cause the therapeutic or the high that people desire, but just 10 times that can be deadly. So anyway, so we will cover that in this chapter. We'll also talk about how these things impact drug interactions. So for example, drug B needs a much higher dose to have its normal effect. And drug A, has its effect at, let's say, this dose. Combining them doesn't necessarily just add those effects together, though. That usually when we combine drugs, you see much greater effects with much lower doses, and often a lot of really toxic effects from drug combinations that would not be predicted by taking either drug alone. Finally, in Chapter 5, we'll go over the principles of psychopharmacology in this module, where we talk about the biological, the psychological, and the social and environmental factors that play into the drug experience. We'll also review in this chapter types of tolerance. I know most people are familiar with tolerance, the idea that we need increasing doses of a drug to achieve the same effects, and that's generally referred to as dispositional tolerance. But we'll also talk about functional, acute, protracted, behavioral tolerance, and cross-tolerance. We'll finally finish up talking about some behavioral pharmacology, that is studying the effects of drugs in animal models. We'll use as an example drug discrimination, and you guys will actually get to read an article that I published as a young graduate student that used a drug discrimination model to study the effects of fluoxetine or Prozac, and how it might relate to two other drugs, cocaine and LSD. And then finally, we'll address some principles of learning and how they affect addiction. Oh, and we will finish up talking about new drug development.